These are the fluidic system, which presents samples to the interrogation point and takes away the waste; the lasers, which are the light source for scatter and fluorescence; the optics, which gather and direct the light; the detectors, which receive the light; and the electronics and the peripheral computer system, which convert the signals from the detectors into digital data and perform the necessary analyses. The interrogation point is the heart of the system. This is where the laser and the sample intersect, and the optics collect the resulting scatter and fluorescence. First, let's talk about how the sample is delivered to the laser. Here we see how the sample is transported through the interrogation point. For accurate data collection, it is important that particles or cells are passed through the laser beam one at a time. Most flow cytometers accomplish this by injecting the sample stream containing the cells into a flowing stream of sheath fluid or saline solution. As you can see, the sample stream becomes compressed to roughly 1 cell in diameter. This is called hydrodynamic focusing. In fact, flow cytometers can accommodate cells that span roughly 3 orders of magnitude in size. In most cases, cytometers will be detecting cells between 1 and 15 microns in diameter, although through the use of specialized systems, it is possible to detect particles outside this range. Now let's see how laser light is used to detect individual cells in the stream. As a cell passes through the laser, it will refract or scatter light at all angles. Forward scatter, or low-angle light scatter, is the amount of light that's scattered in the forward direction as laser light strikes the cell. The magnitude of forward scatter is roughly proportional to the size of the cell, and this data can be used to quantify that parameter. But how can we record this scattered light? Light is quantified by a detector that converts intensity into voltage. In most cytometers, a blocking bar, called an obscuration bar, is placed in front of the forward scatter detector. The obscuration bar prevents any of the intense laser light from reaching the detector. As a cell crosses the laser, light is scattered around the obscuration bar and is collected by the detector. The scattered light received by the detector is translated into a voltage pulse. Because small cells produce a small amount of forward scatter and large cells produce a large amount of forward scatter, the magnitude of the voltage pulse recorded for each cell is proportional to the cell size. If we plot a histogram of these data, smaller cells appear toward the left and larger cells appear toward the right. A histogram of forward scatter data is a graphical representation of the size distribution within the population, but such a graph only presents one-dimensional data.